Topic we'll be talking about is the smooth muscle and all its properties and how it contracts and functions. So when we look at the smooth muscle, it's an involuntary muscle, okay, like your cardiac muscle. It's non-striated, so it's not organized in the way that your skeletal and cardiac muscle is organized, and it's very, very efficient. It can be really efficient and really strong in making and generating a lot of force of contraction. And if we look at the nerve supply over here, there's a picture over here. This is the skeletal muscles, typical neuromuscular junction which we've seen. In the smooth muscle, the neuron that supplies it, these autonomic nerves, usually they're supplied by autonomic nervous system. So the ANS, autonomic nervous system, sympathetic and parasympathetic, they release neurotransmitters. But if you see the structure of this neuron, it's not like a terminal bouton ending and you have the motor end plate over there on the other side, the post junctional membrane. What we have over here is a neuron that is able to have multiple little bulges. You could see over here, these are varicosities. Okay, varicosities are bulges, like when your leg veins get dilated permanently because you stand for a long time and you damage the valves over there, you blow out the valves, so you get varicose veins. Well, like that, you get these dilatations over here in the structure of the neuron that's supplying these smooth muscles that are over here, and we call this synapse in passant, or synapses that are made in passing. And there can be a lot of distance between these varicosities and the smooth muscles. Why? Because it's able to spray the neurotransmitters from quite a height all the way down onto the smooth muscles over there. So it's like how you have those flower sprays that you spray water and it spreads the water from the hose pipe. This is something like that. So it sprays the neurotransmitter on the smooth muscle. They can be filled with neurotransmitters which are excitatory or inhibitory in nature. So you can see over here I've drawn acetylcholine as a neurotransmitter, or norepinephrine as an example over here. Now, smooth muscles, they are relatively less complex. They're not as complex as your skeletal or cardiac muscle, and they need less than 100 times the amount of ATP that the skeletal muscle would need to do the same work. So they are really efficient, and they make a lot of tension, and they generate quite a lot of force. Now, if we classify smooth muscles, there are of two types. Number one is the single unit smooth muscle, which are also known as unitary smooth muscle. They're also given the name visceral smooth muscle because they tend to be around those these tube-like structures like your intestines, GIT, and all of that. So if we look at this, a single unit smooth muscle means that the smooth muscles are there are a bunch of smooth muscle cells, and they're uninucleated, and they are connected to each other via gap junctions. Right, so if you stimulate one and cause an action potential in one of these single unit smooth muscles, then you eventually will create action potentials in all the other muscles because they are connected by gap junctions. So they are in syncytium, just like your cardiac muscle. That's why we call them the single unit smooth muscles or unitary smooth muscles. And they are found in visceral organs like the uterus, the GIT, the ureter, and the urinary bladder. Contrast this with multi-unit smooth muscles or multi-theory smooth muscles. So what do we have over here? We have smooth muscles that perform a function, but they are not connected via gap junctions. They're not connected via gap junctions. So if you release a neurotransmitter over here, then what happens is it will stimulate that particular muscle only, but it will not stimulate the surrounding muscles which did not receive any neurotransmitter. So if we look at the innervation of single unit versus multi-unit smooth muscle, look at this. Multi-unit smooth muscles, you have to stimulate each and every smooth muscle over here. So your synapse in passion will actually be at quite a distance from the smooth muscles, because you want to spray and let all of this neurotransmitter go all over the place. Okay, so you want more distance from the muscle. But if you look at the single unit smooth muscle, if we have a few smooth muscles here, the varicosities of synapse in passion in these neurons, autonomic neurons, they'll be quite close, because if you stimulate one at least, the others will get stimulated eventually because of the gap junctions, because they are in syncytium. So your multi-unit smooth muscles, they don't have gap junctions, so there is no coupling or syncytium. They are seen commonly in the iris, and they determine the diameter of your pupil, and your ciliary muscles again of your eye, determining the length, the focal length of your eye. They control the thickness of the lens. The vas deferens also in the male genital tract, and again you have the piloerector muscles of skin. So these are the smooth muscles that are under your hairs that are present on your skin, and they make your hair stand on end, like when you get the goosebumps or when you're scared. So those are multi-unit smooth muscles. Now if we see the blood vessels, they have single unit and multi-unit smooth muscles in their walls. So blood vessels, they have got both types of smooth muscles in their walls. But in other organs like uterus, GIT, ureter, urinary bladder, it's single unit. In iris, ciliary muscle, vas deferens, and piloerector muscles of skin, they're multi-unit. So let's see the functional anatomy. Mononucleated, and the nucleus is centrally located, connected by adherence junctions and gap junctions to each other. They are not striated, not well organized, and they have the muscle fibers over there, the myofibrils, actin and myosin, but there are some differences. And one more thing you should know, the intermediate filaments of the smooth muscle are desmin and vimentin. Why do you need to know this? Because if you want to know if a particular tissue is smooth muscle origin, what the pathologist will do is look for these intermediate filaments which are unique to that particular tissue. So let's say there's a tumor which you can't differentiate it properly, because tumors are really disorganized. So if you want to know where this tumor came from, or what's the source, what kind of tissue it originated from, you can look at the intermediate filaments. And here in the smooth muscle, it's desmin and vimentin, and we use this in pathology to diagnose what kind of tumor this is, where it originated from. So you have your smooth muscles, they have actin and myosin, but they are different from the actin and myosin isoforms of the skeletal and cardiac muscle. That means you have different part of the DNA, different gene that is coding for this myosin over here. There are no T tubules, they only have these kinds of caveolae. So you could see these depressions which I've drawn over here, these depressions over here on the smooth muscle membrane. These are caveolae, and in these caveolae you tend to have these L-type calcium channels. So they are sort of analogous to your T cells, with the calcium channels located, those dihydropyridine like in the cardiac muscles, and but they're not T tubules. They are caveolae. They're small depressions that are present on the smooth muscle surface, and you can see you have the L-type calcium channels, and you have this 3-sodium 1-calcium antiporter, which are located in these. They tend to be located especially in these caveolae. Now if you look at this one, other differences, there are no Z lines over here in the smooth muscle. They are for anchoring actin thin filaments. They are for the anchoring of thin filaments. So you have the I've drawn over here in the smooth muscle, we don't have Z lines for anchoring thin filaments. Instead, we have dense bodies, which I've drawn in red. They are analogous to the Z lines. So what I've drawn in red is what the Z lines would be in the skeletal muscle or cardiac muscle. It is where the thin filaments gain their origin from. And if we look at this, they contain alpha-actinin. So these dense bodies, which are drawn in red, from which the thin filaments come out, the actin comes out, they are composed of alpha-actinin. And myosin, you could see in green, it's suspended over there. in between these thin filaments, and titin is present to help in organization of all of this. And there is no troponin. Please remember, there is no troponin that regulates the excitation contraction coupling over here. Instead, we have calmodulin in place of troponin, which binds to calcium. So instead of 4 calciums binding to troponin, you have 4 calciums that will bind to calmodulin. And there is no nebulin, which was present on the thin filaments, which you studied about in the skeletal muscle video. Please watch that video before you watch Smooth Muscle. Now, tropomyosin is also present, but it helps in actin-myosin interaction. It does not inhibit the interaction. Remember, in skeletal muscle, tropomyosin used to make sure that the myosin doesn't even get the idea of coming close to the actin over there. So tropomyosin in this case is more like something that's going to help in the actin and myosin interaction. This is a very important point. Tropomyosin is going to help in actin-myosin interaction. We also have 2 other things, caldesmon and calponin. Okay, and these are only present in smooth muscles, and they bind to calmodulin. So what happens is caldesmon and calponin will bind to calmodulin, and this is going to cause inhibition of myosin ATPase activity, causing relaxation of muscle. Because you see, your calmodulin will cause the myosin ATPase to work, and if you look at caldesmon and calponin, they bind to the calmodulin, inhibiting the myosin ATPase activity, causing the relaxation of muscle. Thin filaments, if we look at them, the ratio of the different protein structures are actin to tropomyosin to caldesmon to calmodulin is 14:2:1:1 in smooth muscle. In skeletal muscle, however, it's going to be actin to tropomyosin to troponin in this case, 7:1:1. Right, so you have to remember these numbers. Other than that, let's see the electrical activity of smooth muscles. They don't have a particular RMP. It fluctuates between -40 to -60 millivolts. So it's always hovering around there somewhere. And remember, you don't need action potentials in smooth muscle to cause contraction. What makes the smooth muscle work is the amount of calcium that comes in. So it does not have to have a big action potential and then cause a burst, a spark of calcium to be released into the cytoplasm. It doesn't work that way. Here, you give a little calcium, the muscle contracts a little bit. You give more calcium in the cytoplasm, the muscle will contract even more. So it's another differentiating point from skeletal muscle. So the skeletal muscles do have action potentials, and if we look at the unitary smooth muscles, they have action potentials that are really similar to what you studied in skeletal muscles and in neurons. So they're the typical stimulus artifact, local response followed by spike potential, then repolarization, and all of that's the same.